Hi friends, I am your Karthik Kate and this is Kate Tutorials, right? So in the upcoming videos we are only going to discuss about these alkaloids. So here I have written the definition of alkaloids, what are alkaloids and how we are going to make use of them and everything that we shall see in this video, right? So these alkaloids are nothing but they are always derived from plants. So these plants contain these alkaloids, right? From there we are going to extract. Why we are going to extract? Because these alkaloids have many medical importance, so we are going to extract these alkaloids from plants, right? So what are these alkaloids? In most of the cases all the alkaloids contain this nitrogen, right? So this nitrogen, most of the times these nitrogens will be basic, but there are so many alkaloids in which nitrogen act as neutral. In also some of the alkaloids the nitrogen even act as acidic. How it acts as acidic? Because nitrogen attached to hydrogen, that proton can be removed as H plus. So many nitrogens are there in which nitrogen acts as acidic in alkaloids, right? So all the alkaloids contain nitrogen. So initially scientists what they thought was this nitrogen, always the plants contain alkaloids and all alkaloids contain nitrogen, and they thought that, so they studied few alkaloids and in every case they saw that nitrogen was in the ring, as I have shown it here. Here also in the ring, nitrogen is in the ring, right? Here also in the ring. So they concluded that alkaloids, they contain nitrogen and all nitrogen will be within the ring only. But later they discovered so many alkaloids in which nitrogen was present in side chain. For example, here you can see Physostigmine, in which one of the nitrogen is in the side chain, right? So later they found out so many alkaloids in which nitrogen was present only in the side chain, not in the main ring. So they concluded, so alkaloids are nothing but they are present in plants and they contain nitrogen, and they didn't mention whether it is in the ring or side chain, right? So that is the thing. And also they told that those nitrogens, most of the times so many alkaloids have basic nitrogen, but there are few exception in which nitrogen are neutral and some are acidic also, right? So that is the definition for these alkaloids, right? So these alkaloids, they include many drugs. For example, morphine, quinine, we can use them for medical purposes, for severe pain. If people are suffering in pregnancy or in some accidents, to get relief they give these drugs, right? So but the thing is, if normal people take this drugs, what they will do, they will get hallucination. To do the nasha, if they take these alkaloids, then it becomes illegal, right? So many countries bans this things. So we should only use for it medical purposes only, right? So in hospitals they will make use of it, right? And also so many plants, they will tell, if you go to any forest and anywhere without knowing about plant, you should not eat any of the thing. We should not eat their leaves, bark, seeds, fruit, because they contain poison, right? Because so many alkaloids are poison. For example, atropine is the alkaloid which is poisonous, right? Similarly, morphine and quinine, these are used as drugs. And also you might have heard about LSD, right? So LSD, this is nothing but this LSD contains lysergic acid. So this is also used as a drug only, right? So many people misuse it, right? So LSD sticker which they will attach in the mouth, so many things they will do, so many nashas, right? Sanju movie also you can see those things, right? So these are the alkaloids. Why I have written these alkaloids? Because we are going to study these alkaloids only. So this is the syllabus what we are going to study in alkaloids. So what we are going to study? We are going to study the Papaverine. So structure elucidation of Papaverine means initially we don't know the structure of this Papaverine. How we are going to get this structure only, that how the scientists did, how they carried out the reaction, what are the chemical steps they involved, what are the chemical degradation methods they did, so many things, and then finally they concluded this product. Once they get this structure, this Papaverine contain only this structure, then they will independently synthesize this within the lab. So how they are going to do all those things, that I am going to explain it step by step in the upcoming videos. So here we are going to study the structure elucidation of Papaverine. So Papaverine, its structure I have written here, so you should remember it if it is there in your syllabus. Similarly here morphine, you know morphine, It is also famously called as heroin, right? So many, it is used as a drug which is illegal, so it is banned in most of the countries because it is used as a drug only, right? So this is the structure of morphine, and we are going to study the structure elucidation of morphine, how we are going to get this structure by so many methods, everything, chemical degradation, everything we shall see, right? So Reserpine, so Reserpine structure is this one. So these things I am going to explain in detail in the upcoming videos, right? So structure of Reserpine, and you can remember all the structures because even I have done my MSc, I have completed, right? So even I have remembered these things, everything, because the only thing to remember is not to by heart. Just you see the structure, you will get to know. If you watch my upcoming videos, you will see that how I am going to link it. After linking, like all links, if you get it, if you know the chemistry, then it becomes easy to remember the structures, right? So it will happen, how it happens, everything you will see in the upcoming videos, right? So this is also Physostigmine. It's one more name is this Ezerine. So Ezerine or the Physostigmine, and its structure is this one. So it contains 3 nitrogen here, 2 of them are in ring and 1 of the nitrogen is in side chain, right? So this is the structure of Physostigmine, right? This LSD, it contains this lysergic acid. This is the structure of lysergic acid. If you want to know the structure of LSD, just nothing to do. This acid group is there, right? Just you remove it. You make it as C double bond O. That OH you remove and it is replaced by one N which is attached to ethyl CH2CH3. So this is the structure of LSD. LSD means lysergic acid diethyl amide, right? Because this NCO bond is nothing but amide bond. So what is this LSD? Lysergic acid diethyl amide, not amide, right? So that is nothing but our LSD. But for our study, we are going to study only lysergic acid because this thing we can easily synthesize, right? Once we get this lysergic acid, which contains COOH, we will get that LSD, right? So these are the things we are till this lysergic acid we are going to study only structure elucidation, means we are going to find out the structure, and also we will synthesize it, how the scientist did, that we will discuss. But for these compounds, these 3, Tylophorine, its structure is this thing, Nuciferene, its structure is this one, and this Corydaline, its structure is this thing. So all these we are going to synthesize them, not to structural illustration, we are going to synthesize that to photochemically using light radiation, how we are going to synthesize this compound, that we are going to study. So about all these things I am going to explain in detail in the upcoming video, so no need to worry. So this is the introduction class, let me give the introduction, right? So this is about all the things which I am going to tell, so you can take it as a content, right? So once we discuss this, so what? So we define the alkaloids, right, their uses, and also we saw few of the alkaloids and I have written the examples, right? So you can make use of it, right? So about LSD we have discussed. So I got something but I forgot to tell, no problem, right? And these things and these alkaloids can be divided, means these are classified as so many things. See here, based on the structure only we can classify these alkaloids. How we are going to classify? See, if you see this structure, what it is, it is nothing but isoquinoline. Isoquinoline means, since this papaverine contain isoquinoline skeleton, so this is also called as isoquinoline alkaloid. Like that they are going to classify. For example, if you come to this LSD or this lysergic acid, you will see this skeleton. So what it is? This is nothing but indole. Benzene ring containing a pyrrole ring, that is nothing but indole. So this is classified as indole alkaloid because it contains indole skeleton. In the same way, all these alkaloids are classified differently. Some are classified as pyridine or hydropyridine alkaloid, isoquinoline alkaloid, quinoline alkaloids, and this is indole alkaloids. So many things. This Reserpine, so it also contains this indole skeleton. So we can call it as indole alkaloids, right? So many things, right? And here if you see this, coming to tylophorine, if you see, this is what, this is nothing but the skeleton of phenanthracene. Anthracene, you know, this is anthracene, right? This is anthracene, right? This is anthracene. If I remove this last ring, instead of taking this in same line, if I take it here, right, then it becomes phenanthracene, right? So this is nothing but phenanthracene alkaloid, tylophorine, right? So in this way, based on the skeleton, based on the structure only, that to contain the main skeleton, they are classified differently. So they are classified as isoquinoline, quinoline, so many indole, this phenanthracene alkaloids. So like this they are going to classify it. So what all the things we are going to study in the upcoming videos. See, so let me rub those things so that I can give you the idea what we are going to study in the upcoming videos, right? So you can see that, see, always whenever we do this alkaloid, so we will always do the structure elucidation. So if I have any bond like this, if I do ozonolysis, what happens? This bond will be broken. So what I will get? I will get H, here double bond O comes, and here CH3 double bond O, acetone. So what we do, when we have any alkaloid, if I take, which has this double bond character within it, if I treat that alkaloid with ozonolysis O3, I will get one of the product. If I get acetone, we will conclude that it is having CH3 attached to double bond, like that. So this method we call it as chemical degradation. By this method we can tell that whether double bond is there or not. Like that we are going to do so many reactions, like oxidation reactions, in which we are going to use KMnO4. So using this oxidation we will get different product. So we can tell why we got this product. So this product is having ketone group, then it contains something else here, secondary alcohol. We can tell like that. So with KMnO4 degradation we do, ozonolysis degradation we do, so many reactions we will do to tell the structure of this alkaloid. That you will see in detail when I take each alkaloid and I explain the structure elucidation. That you will see in the upcoming videos, right? So about that we are going to discuss. And also, if I have any RO6, if I write like this, it is understood that here oxygen is attached to CH3 group. So we always write like RO, like simple line means CH3 only. Sometimes we write RO, Me means methyl. Methyl is nothing but CH3. All are same only, right? You should not get confused. Similarly, sometimes we write as ET. ET means ethyl. Ethyl means CH2, CH3, right? Or sometimes we will directly write ET or CH2, CH3, or sometimes we even write C2H5, or sometimes we directly write like this, CH2, CH3. If I have RO, CH2, CH3, right? Simply we will write like this. So these are the things you will know. So I should not tell, but let us keep it simple, right? So this is the thing, right? So whenever we have like OCH3 group, OCH3, let me take this RO methyl. If I treat with HI, I will get ROH plus methyl iodide. Simple reaction. The H will attack here by taking its lone pair, so it gets positive charge. This I minus will take this methyl, so it will form CH3I, methyl iodide, right? So we can estimate methyl iodide, based on which we can tell whether this alkaloid contain how many number of ROCH3 groups. Means OCH3, number of OCH3 we can tell. For this compound, if I treat with HI, it will consume 4 equivalent. It means it contains 4 OCH3 groups. You can count here 1, 2, 3 and 4. So it consumes 4 equivalent of HI to give 4 equivalent of OCH3. So we measure this number of equivalent, how much we are getting in the product. If you are getting 4 methyl iodide, it means it is consuming 4 equivalent of HI. It means what? It contains 4 unit of OCH3 within a papaverine. So we are going to take only one equivalent and we are going to react, but we are getting 4 methyl iodide. It means it has 4 CH3 group. So like this we are going to tell the nature of OCH3. If you can see here if I remove this methyl, if it becomes OH, right? So OH means here it will be like phenolic OH, not alcoholic, because OH is attached to aromatic ring. So if we get any phenol, if we treat with FeCl3, neutral FeCl3, we know it will form a charged complex that is violet in color, because it is going to form Fe, this is C6H5O minus 6 times something charge will be there here, coordination complex it is going to form, and the color of that complex is violet, right? In lab also we will do quantitative analysis, right? Qualitative analysis. Qualitative means you will tell whether phenol is there or not using this FeCl3 only, right? So that method we can check whether phenol we are getting or not in the alkaloids. So we will conclude that if violet color comes, then the alkaloid contains phenolic OH. Like that we can conclude so many reactions. So these are the reactions we make use of, right? And also we can tell the nature of nitrogen in the alkaloid. So let us assume that this is my alkaloid which contain nitrogen. So if I add CH3I, if I get quaternary ammonium salt, it means what? This alkaloid is there, it is tertiary. Since it is tertiary, if it reacts with one molecule of CH3I, it will become quaternary, because CH3 now nitrogen positive, it means all the nitrogen is attached to alkaloid only, so it will be quaternary. For example, here you can see this nitrogen is tertiary. If I attack with this CH3I, it will form like this structure, CH3 plus, so quaternary ammonium salt. So using that we can tell whether the nitrogen is primary, secondary, tertiary or not, right? If it was secondary, what happens? Secondary means nitrogen is attached to any of the RR here, one hydrogen. It will consume one equivalent of CH3I, it will become tertiary now, tertiary. Further it will consume one more equivalent of CH3I, then it will become quaternary, right? So what you will understand? So since here it is forming salt, so it is forming quaternary ammonium salt here, then it means here it is tertiary and here it is secondary. So the alkaloid containing nitrogen is secondary. Like that we tell the nature of nitrogen. So nature of phenol we can tell, nature of OCH3 we can tell. So many reactions which you will study in the upcoming videos. Like this we are going to discuss, right? So I hope you understood. This class is only for introduction. So what all the things we are going to study. When you see the things, the upcoming videos, there you will get how in detail we are going to discuss about this alkaloids, right? So now let us see how we are going to extract this alkaloids, right? So I have one alkaloid. It is present in plants. So plants means it has got so many things. So leaves are there, right? Leaves are there, seeds is there, fruit might be there, roots are there, stem is there, so many things, right? So the alkaloid can be present in any of the thing, right? For example, if you see caffeine if you see, it is present in tea leaves. So in lab during MSc, you will extract this caffeine from tea leaves. That you will learn in MSc labs only. We will take this tea leaves, we boil it, we add some chemicals, so that we are going to finally extract this caffeine. Once caffeine comes, we are going to do spectroscopy like IR, NMR will remove, so that we will confirm that whether we have got caffeine or not, like that. So these plants, anything, let us take this leaves. So leaves we cut into small pieces, or seeds we make them a powder, right? So that we will convert it into powder form. Once they are dried and powder, then what we add some like organic solvents, because if you see, I had here written so many things, all were containing carbon, hydrogen, nitrogen only, right? So all those are non-polar. those are not soluble in water, right? So they are only soluble in organic solvent. So most of the times we add methanol, so that it is having low boiling point, it can be easily removed, right? So we will extract it. So from extraction we will get within, will take this thing and we will keep it for drying, so that methanol can escape and we'll get the solid extracted alkaloids, right? So that is how we will do. So solvent extraction we will do, in which organic solvent we will use methanol. So the solvent extraction, it may takes weeks, right? So many things, for 48 hours, more than that we will keep it, so that we will extract all the alkaloids. Once this methanol is there, then it is treated with some inorganic acids like HCl, perchloric acid, HClO4, right, acid, to get the salt. Once we get salt, then we will analyze it. So many chemical methods we can analyze it, right? So we analyze. So this is how we are going to extract. You can see that plant, if you take plant, we will take in bulk. But the thing is, if this is the plant, only this much of alkaloid is present within that plant. So the percentage what we get of the alkaloid will be very less, right? Because alkaloids present in that plant will be very less. So that is the problem. So here, once we got that product also, now we have like TLC plates are there. So we will this is the TLC plate, reference line we will add. So these are the compound we can place here, and we will run the solvent here. So we will see that solvent has ran here. So we may get like two like this. So it means our compound which we have extracted, it contains two kind of compounds like this. Like this we can purify them, like, right? So like this we will do, so that we will extract the pure alkaloid. So these are the things, right? So this is how we extract the alkaloids. So I hope it is useful for you. So this was only simple introduction in which I told you the content. So in the upcoming classes or the upcoming videos, you will see only the things which we are going to discuss. That is means one of the compound we are going to take, and A to Z of that compound we are going to discuss. So you please watch those videos. If you like, please give me a like button, and please share with your friends, and take care and have a great day.